Okay, good morning. Let's talk a bit more today about the structure of how our DNA is organized inside of our cells. Um, so remember, we've been talking the last few lectures about the importance of polymers. I think sometimes it's easy as a student to kind of lose sight of sort of the big picture of why we talk about certain things, especially when it comes to particular types of molecules, particular arrangements of molecules. I know when I was a student taking cell biology, my professors would teach us about different polymer structures and things like that. And in a way, it's kind of like, yeah, I understood that these molecules, are these polymers, but I didn't really understand or appreciate why kind of knowing about that was important. Um, I think the one of the easiest ways to kind of have a perspective on this is, of course, living things like the individual cells that make up our body are composed of a wide range of different types of molecules that over the course of evolution were selected for because they serve a purpose, they serve a function. And many of these molecules serve their function just as small little molecules. So many types of nutrients, for example, many types of other important gases and things like that, oxygen, CO2, they just function as small molecules. But you also have, in the course of evolution, right, the dependence upon certain arrangements of small molecules of a particular type into long, long chains. Right? And as a long chain, you actually have certain benefits. For example, a long polymer of simple sugars to make a polysaccharide is a nice way to have many energy-containing bonds in one molecule that can be handled very easily. Um, Lipids, for example, they're not polymers, but this idea of long chains of bonds that you can get energy from, right, is a very important part of lipids. Lipids are also parts of important structures. Speaking of structures, proteins, which are long polymers of different amino acids, right? We learned that there are 20 amino acids that life on Earth depends upon. That is a great way to build complex structures, right? Either as a long chained polypeptide that could be a protein that's meant to be a long chain, so structurally that's useful, or as we discussed with proteins, and we'll talk about a lot more, the important thing is when those chains of amino acids are folded up in particular ways, you get three-dimensional shapes, complex three-dimensional shapes that give you lots of opportunities to have specific shapes that can fit together with other shapes like other proteins and function as little machines. And of course, long polymers, if we're talking about chains of nucleotides, A's, T's, C's, G's, and sometimes U's, right? Uracils, we're talking about RNA. That is a fundamental way of storing information, right? The sequence of nucleotides, right, is not random, right? It is a particular sequence of nucleotides that represents a message that our living being depends upon in the form of all the different genes that make up our genome. So I think when we discuss things like complex macromolecules, I think it's always important to keep in mind, right, that these things come together because of life. It is our living cells that put together things like polymers, right? And at the same time, these different polymers perform different important functions that our cells depend upon. So last time we left off talking about the structure of DNA. Right? And we looked at how a little bit of the history of how Watson and Crick, using the data that Rosalind Franklin generated on the X-ray crystal structure of DNA, came up with the first correct structure of DNA, the double alpha helix. And I mentioned to you that when you look at the structure of DNA with the base pairs, the ribose or deoxyribose sugar rings, and the phosphate backbone, I think it's very important that you recognize that and understand that and are able to really see how those different pieces come together. Oftentimes students kind of ask, oh, why do I have to know the structure of DNA and being able to, draw it out or recognize where all the different connections are between the nucleotides, the bases, the phosphodiester backbone. I think it's easy to think, oh, it's just busy work, right? But it's very significant if you're going to study life to be familiar with that. So let me tell you a little story to maybe help shed some light on why I think that's so important. Okay. I'm kind of prone to telling stories. So this story has to do with me. So that's me back in 1977. Okay. So I think this is a photo of maybe like the first day of, I don't know what it is, maybe kindergarten or something like that. Okay. And so this is me in front of the house that I grew up in Massachusetts, because I grew up just outside of Boston. Um, and I like this photo because it reminds me of the house and it makes me think about, what it was like being a kid um, growing up outside of Boston. Every maybe a couple weeks or so, my folks would drive with me into the city. Right. And I loved this city. I thought it was a really exciting place. But I remember as a kid, my parents taught me this very important lesson. They said, if I ever got lost in the city and separated from them and couldn't find them, that I should try to find a nice policeman or policewoman and tell them I'm lost and give them this information. Right. 145 Richdale Road, Needham, Massachusetts, 02194. Because that was my home address. Right. And my parents taught me that if I could remember this, right, then that police officer could help me get home. Keep in mind, this is well before things like cell phones, right? So this was what my parents thought was the safest way. I needed to know this, right, in order to get home safely. And as a kid, I remember sitting there at the kitchen table looking at this. In kindergarten, I had my writing wasn't that great. I couldn't really spell a lot, but I tried to commit this to memory, right? Because I thought it was kind of weird, this combination of numbers and letters, right? That really are just letters and numbers, like some sort of code on a piece of paper. But if I could know this, right, it had a lot of significance and meaning because this is where I belonged. This was my address, right? So it's very important that I remembered it. So I did, right? I remembered it. I'd memorized it, how to write it out. So why do I say that? Okay. Because there's another important address that we've been talking about in this class, right? And that's this. So this you can think of as just a whole bunch of numbers and letters, right, put down on a piece of paper, drawn in a certain way. And you can say, yeah, it has meaning because this is, our DNA. But have you ever thought about the importance of why, as a life science student, you should know this well? Right? This particular address, because it's an address to somewhere specific. And that's this. This is our home, planet Earth. Right? All life on Earth uses this address. This address, as far as we know, is unique to planet Earth. I've already shared with you this kind of broader way of thinking that in reality, look, the DNA molecule that you see here, it is just atoms, right? Atoms, nitrogens and oxygens, phosphates that are all linked by carbons to form these different structures. Right? Certainly, these atoms and different parts of this structure exist throughout the universe. Right? And I said, throughout the universe, there are a bazillion other variations of things like nitrogenous bases, right, or sugar rings, right, things like that. But the important thing to keep in mind is that as far as we know, only on planet Earth did life evolve to depend upon this very specific arrangement of atoms and small molecules to form genetic information in the form of DNA. That means if one day, for some crazy reason, you take a wrong turn as you're driving home and you find yourself, a few light years away from Earth, off some other part of the Milky Way galaxy, right? And you're lost. Okay. And then fortunately, the spaceship kind of flies by and it's some advanced alien civilization, right? And they take you in and they say, "Where are you from?" Right? And then you're like, "I'm from Earth." And they're like, "What's Earth?" Right? Maybe this advanced civilization, they've been, patrolling around the galaxy, cataloging the different genetic codes, the different molecules that make up life on different planets. If you can draw this, right, then perhaps they would say, "Oh, we recognize this. We know what planet you're from because all life from that planet shares this same address." Okay. And they're like, "Well, we were going to head over there anyhow because there's another one of you Earthlings on board. So you guys might as well travel together and we'll take you home." Right. And you're like, "Hey," and they're like, "Okay, yeah, you can share a room with them." And then you're like, "Well, I don't see them." And then you look down and there's like a slug or something on the ground. Right. And you realize, "Oh, this is my fellow Earthling." Right. And the slug looks up and says, "Hey, what's up? Yeah, we're going home." Right. Because the slug has this same genetic code, the same system of DNA, right, that identifies you both as being uniquely life from Earth. Right? So if you're a life science student, whether you're going to study botany, animal physiology, marine biology, right, how can you deny that at the very core, all of those living things that you'll be studying, right, share this as their unique address that makes them an Earthling? Okay, so that's why I feel it's important that you're able to recognize this structure. Let's talk today a bit more about how DNA is actually packaged up inside of our cells. DNA is a tremendously long polymer. So, how physically long is the DNA of a teeny tiny organism? Some of the smallest living things on Earth are, of course, prokaryotes, things like simple bacteria. Mm. So maybe we can start with thinking about how large is the DNA molecule of a teeny tiny bacteria. So a bacteria such as E. coli is about 2 micrometers long. So if you look at these rod-shaped E. coli bacteria here, all right, this is 2 micrometers long. So that's really small. That's maybe like the 10th or even smaller than a typical cell inside of your body. All right, so the DNA inside of it is sort of loosely um, present. Oftentimes you'd find loops. The genome is in a loop that we call a plasmid, which is a circular strand of DNA, and it's contained in a sort of nucleus-like kind of compartment, which isn't a true nucleus because there's no membrane around it. We call that a nucleoid. Right? If you were to unravel the genome of an E. coli bacteria and actually straighten it out, right, and measure it from one end to the other, you'd find that the plasmid is about 1,600 micrometers long. In other words, the DNA alone is 800 times as long as the bacteria is. This image on the right here shows a bacteria that has been lysed, and you can see its genome is spilling out. Right? So it's remarkable that humongous molecule is packed into a small space. And that's just a little bacteria's genome. Our genome is much larger. So how long is human DNA? If you were to take from your body, your body is made of trillions upon trillions of cells, right? I mean, trillions, I say trillions, but that's like almost meaningless because I can't grasp in my head what a trillion is, right? Yeah, 1,000, a million, a billion, a trillion. At that point, it's like whatever, right? If you were to take one single nucleated cell from your body, let's say you take a skin cell from your arm, okay, and you were to lyse that cell, okay, and unravel your genome that's contained in that cell. Now, of course, our genome is not just one single DNA molecule like the bacteria is. Our genome is present as 22 pairs of autosomes, so 22 pairs of single molecules, plus a pair of sex chromosomes, right? So each one of our chromosomes is an individual DNA molecule, right? And we have numerous chromosomes. That's the way our genome is arranged on separate molecules, right? Which again, as a undergraduate, I never really thought about that and how strange that is, right? That we actually have separate molecules that together represent our entire genome. But if you were to then unravel each chromosome and line them up end to end to end to end, right? Then you could say this is the complete human genome. This is the complete genetic message that codes for me. And how long would that be? Right? The entire genome, a single copy of which, because remember, you get one from mom and you get one from dad. If you were to line it up, it would be 6 feet long. Right? That's remarkable because you think about it, 6 feet long, right, is taller than most of us, right? And yet you would not be able to see it because it's so tiny, right? Because it's just one double helix of DNA, right, from one end to the other, right? You would not be able to see it with your eye, and yet this thing is 6 feet long. So the incredible thing is, how can 2 copies of that, one that came from mom, one that came from dad, fit inside of a cell from your body that the cell itself is maybe only like 5 micrometers in diameter, right? So very clearly, the DNA must be highly organized and tightly packed. So this is an example of what we call a karyotype, Karyotype. A karyotype is when people like in the clinic, for example, for the purpose of genetic testing, will take samples of a patient, of a subject, and try to find examples of cells under the microscope that are in the process of mitosis. In other words, the DNA, the chromosomes have condensed as part of getting ready for the cell to get ready for mitosis to divide. And they will take those cells and they will stain the different chromosomes in that cell, right, with different types of molecular markers that fluoresce in different colors when sort of irradiated by certain types of wavelengths of light. And then they will take images of that, and then through a photo editing type of software like Photoshop or something, actually cut and paste and arrange the chromosomes that they see in this type of karyotype image. And so you can see here the chromosomes, each one of which is a single DNA molecule. It's typically arranged from the largest chromosomes, like chromosome 1, down to the smaller ones. So you have 22 pairs of these. Again, for each one of these, like chromosome 1, let's just pretend the left one came from mom, the right one came from dad, right? And then in this case, right, mom gave you an X chromosome, because that's all that mom can pass on to her child, right? And dad gave you an X chromosome instead of a Y chromosome, right? And so that's why in this particular view, this is a female. Right? So imagine that though, right? We're talking about 6 feet worth of DNA, okay, or technically, I guess you could say 12 feet worth because this is both copies from mom and dad, all packed into the nucleus of a teeny tiny cell. Right? So one of the first levels of organization of how the DNA is arranged, because clearly, and I think all of you know this, our DNA is not floating around just as naked double helical DNA inside of our cells, right? it has to be organized. So it's tightly associated with different proteins. And these proteins provide a way of structurally packing the DNA up so that all of that DNA, that huge molecule, can fit in a teeny tiny space. And importantly, the association between the DNA and the proteins that pack it up provide a way of controlling how different parts of your genome at any given time, any given moment, are a bit unraveled to allow the genetic information contained within the DNA to be read. Okay. So eukaryotic DNA is wrapped around structures at the lowest kind of level called nucleosomes. And each nucleosome is an octamer, meaning it's made up of 8 separate histone proteins. Okay. In this figure here, that's what these purple spheres are. And that means each nucleosome is made up of 2 each of these histones. So 2 H2As, 2 H2Bs, 2 H3s, right, and 2 H4s. And that forms this here, the core of the nucleosome. In addition, it will have 1 histone H1 represented in green that kind of helps keep this nucleosome together and helps keep the DNA kind of wrapped around it. You can see the DNA, the double helix, on the cartoon here. It's coming from the left, and it kind of wraps around the DNA, right? Wraps around the nucleosome here. It's kind of held in place by the H1, and then it continues on through sort of this naked stretch of DNA to the next nucleosome. So each nucleosome is about 10 nanometers in width. If you were to count the number of base pairs, individual like A, T, C, and G rungs of the ladder going around this, it'd be around 146 base pairs of DNA encircle one nucleosome. Right? This is what we call the sort of beads on a string arrangement. So this is a really cool high-powered electron micrograph showing the DNA wrapped around these nucleosomes. You can very clearly see each nucleosome here, around each of which would be 146 base pairs of DNA. And then you have this variable length kind of naked DNA that kind of connects in between. An important thing to keep in mind is that this is not a super rigid, fixed in stone structure. This is a dynamic structure. The proteins, the purple histone proteins that are interacting with the DNA, they're doing so not by covalent bonds that like tightly anchor it, right? This all has to do with shape. Again, the importance of shape, right? So that means these histone proteins, as they're kind of grouped together to form a nucleosome, they will have 3-dimensional surface features that represent places where the double helix of the DNA can very comfortably, very easily fit in, right, to help wrap around. And that means it's dynamic. These nucleosomes, as a result, can move kind of right and left. If you have 2 nucleosomes together, like the 2 purple ones you see here, if one moved to the left more and one moved to the right more, right, then the amount of naked DNA in the middle would be extended, right? More and more of that DNA would be revealed, okay? Or if a nucleosome itself fell apart and the histone proteins no longer stuck together, right, then whatever genetic information was contained in the DNA that's wrapped around that particular nucleosome becomes available. So the lowest level of organization is this beads on a string or pearls on a string arrangement. Okay, so you may notice that these histone proteins, the way they're drawn, have these kind of squiggly looking little tails sticking out of them, right? So what these are, remember, even though these histones are shown as these purple balls, in reality, each one is a protein. So each one is its own polypeptide chain of amino acids that, when folded up, formed this, well, the cartoon shows it as a ball, right? But that means you're going to have the 2 ends of the chain. Maybe one end is tucked in there somewhere, right? The other end, in this case, the amino terminal end of the histone proteins, are kind of sticking out from the main ball of the protein. This is important because that little bit of extra amino acid sequence, right, can be the target of different types of modifications. The addition of different small chemical groups to the amino acids in that part that's sticking out can alter the shape of the histone. That's what this cartoon is showing here. So again, here are the histone proteins. Each one has its amino terminal end of the chain kind of unraveled a little bit, sticking out from it. And certain amino acid residues on these histone tails can be modified. For example, you can add small R groups to them, such as methyl groups, CH3, or acetyl groups, just as examples. So this cartoon here shows certain amino acids might be methylated. In other words, you have the ability, or your cell has the ability to add methyl groups to them, or acetylated. Okay, so as a reminder, a methyl group looks like this, an acetyl group looks like this. And even though something like a methyl group is small, it's just a carbon with 3 hydrogens on it. Remember, don't think of these as stick figures, C's with 3 H's. Think of them more as these sort of little plastic balls that represent each of the atoms. And remember, I've been trying to emphasize to you thinking about this as a 3-dimensional shape, right? That 3-dimensional shape may be enough to cause the histone protein tail, right, once this methyl group is put on there or an acetyl group is put on there, to kind of have to adjust its shape because of the other amino acid side chains that are sticking off on it. It's going to kind of have to shift around a little bit, and there will be kind of like a ripple effect, right, that will cause the main purple ball part of the histone protein to also have to kind of adjust its shape, and that may have a consequence that might lead to the contact between the histone protein and the DNA double helix being wrapped around it, either becoming more tight, right, in which case the nucleosome is tightening its grip on the DNA, or it could be the opposite, right? It could cause the shape of the histones to change so that the DNA doesn't wrap around it as well, and then the DNA will more easily unwrap from the histone. So this is what we call the histone code. Right? Down below is a wonderful figure that kind of annotates many of the particular locations of different amino acids in the histone tails that we now know when they are modified through methylation or acetylation. Sometimes they can be phosphorylated, right? A PO4 group gets put on, or small proteins can even be added, like ubiquitin. That's what this UB is. Right? You can see many of these are lysines. Oh yeah, hopefully you've been making some progress in memorizing your amino acid abbreviations, right? K is lysine. So many of these are lysines that get acetylated or methylated, and that can impact the shape of the histone protein and how it has to shift around. So typically, histone acetylation leads to looser packing of the DNA around the histone, whereas histone methylation typically is associated with tighter packing of the DNA around the histone. Okay, and as I said before, this is a very dynamic process. As you're sitting there, viewing this lecture, right, in each one of your nucleated cells in your body, your whole genome, is being which is wrapped up around things like histone proteins, right, is in a dynamic state of different parts of the histone tails being modified. So certain histones are loosening their grip, certain nucleosomes are tightening their grip. So your DNA is a dynamic, changing landscape of activity. So here's another view. If we were to look at a nucleosome core particle again, and as a reminder, it's made up of an octamer, right? So if you looked at these colors here, these 4 that make up the octamer, these histones, so you have 2 blue ones, 2 green ones, 2 orange ones or yellow ones, 2 red ones, right? That forms the core of your nucleosome. So in this case, we're looking at the DNA wrapped around it, not really coming in from the left and then going off to the right, like the cartoon showed it before. We're kind of looking at sort of head on now, right? You can imagine that this is looking down sort of the DNA wrapped around, right? It'd be 146 base pairs of DNA. I cannot really tell. I assume this is one end of the DNA kind of coming in. I'm not really sure where the other end is. Maybe it's over here, right? Because you're looking at it in 3D spiraling around. But you can appreciate how the structure of the histone proteins forms this nucleosome, right? And you can already tell just by looking at the ribbon diagram of these histone proteins, you can see how the shape is conducive for the DNA to wrap around it. And importantly, you can see that each one of these histone proteins, the N-terminal part of it is kind of unraveled, and that's what sticks out. And that's where you would have certain amino acid residues, like lysines, for example, that can be post-translationally modified. Okay? So this is a nice kind of structural backbone model. But remember, I always like to think about things in three-dimensional space, right? So what if we filled in now all the electron orbitals around these, right? So taking into account all of the amino acid side chains that would be coming off of this peptide backbone and all of the atoms that would be representing things like the phosphodiester backbone of the DNA, then it would look something like this, right? Wow, now you can really see a couple of important features, right? Number one, you can really appreciate the fit of the shape of the histone proteins that make up the nucleosome and the double helix of the DNA around it, right? And you can appreciate, again, this is not forming covalent bonds, right? This is simply shapes fitting together like puzzle pieces. And depending upon how these tails of these histones are modified through methyl groups being added to them or acyl groups, right? If you add certain groups to this, it will change its shape just because the amino acid side chains that are represented here, right, get kind of crowded and they'd have to shift around, and there'd be a trickle effect that would affect how the histone protein inside the core would also kind of change its shape. And maybe that would loosen up its grip on the DNA or tighten it, right, depending on what modification is being made. I do want to say you do not have to memorize the histone code, you know, on that previous two slides where it has all the different locations of these. I don't consider that important. I consider it important that you understand the significance of it and why currently there's so much research into sort of deciphering this histone code, right? Because by understanding how different histone modifications on the tails that stick out of the nucleosomes function, right, you have a better understanding of how the cell is actually dynamically controlling the state of the DNA. Okay, so let's talk about levels of organization, right? We started off with if you just have the naked DNA in its double helix, we have it kind of wrapped around nucleosomes here, right? So we have your beads on a string. So this image over here, this is kind of neat, right? This is about as zoomed in as you can get. This is just the naked double helix. It's only about 2 nanometers in width. But once you put it on the beads of the string, so wrapped around the nucleosomes, it's about a 10 nanometer thick strand. And that's just the diameter of the nucleosomes. So the next level up then would be if you took those beads on a string on the top here, and then you started kind of winding them around each other. I often think of this like a rubber band. If you have a large rubber band, right, and you take it between your 2 fingers, I mean, we've all done this, and if you're bored, you just kind of twirl your fingers, right, and the rubber band begins to twirl up between them. I think we all know what happens. As you twirl, it begins to kind of twirl on itself, right? It gets bunched up on itself as it gets kind of tighter and tighter and tighter. So that's the next level of arrangement of our DNA. It's the 30 nanometer, what we call chromatin fiber. What is chromatin now, right? Because very clearly, this is no longer naked DNA. This is DNA that's tightly associated with different proteins that are meant to organize it and control how the genetic information is read, right? So that complex arrangement of DNA with all the proteins that help organize it, that's what we call chromatin, right? So in other words, the DNA inside your cells right now, right, is packaged as chromatin. So this is the 30 nanometer chromatin fiber, right? So if you say that that part right here is zoomed in right here, right, you can also see that the whole thing can kind of form loops to condense even more. Right? So you're really beginning to package it tighter. So from loops, as seen up here, again, the loops themselves can be made to sort of wrap up on itself, right? Getting more and more kind of compact. And you get this 700 nanometer fiber, which we call heterochromatin. Right? Heterochromatin really suggests that by now at this level, the individual base pair sequence of the DNA that's involved here, right, is really hidden, right? Because it's so packaged up now, first wrapped around the nucleosomes, and then the nucleosomes kind of all wrapped around themselves, and then that then wrapped upon itself. It's so tightly packaged that the genetic information is no longer visible, right, to other things inside of the cell. So regions of our genome, of our chromosomes, that contain genes that are not being actively used or actively being read, you can expect that those regions of our chromosomes would be packed up very tightly as heterochromatin, whereas other regions of maybe the same chromosome, where there are genes that need to be used at any given moment, right, would be more loosened up. They'd be more like the loops that you see at the top up here. Okay. So that means most of the time in a cell in your body that's doing its job, right, functioning as like a skin cell or a nerve cell or muscle cell, or gland cell, its DNA is going to be arranged as a mix of heterochromatin and just sort of more relaxed loops, right? And that means your individual chromosomes are not going to be present as like these rods that we usually see in a karyotype, like the rainbow colored image I showed you earlier, right? Because those condensed rods only happen when the cell is about to undergo mitosis. So that means during mitosis, that's when you see the next higher level of condensation, right? So now the DNA is so condensed, right, that it looks like this typical rod shape, right? Or in this case, it looks like an X. Why does it look like an X? Because as part of mitosis, you know this, right? Each one of your chromosomes has to replicate. So you have two copies of the same chromosome, and eventually one copy is going to go to one daughter cell. The other copy goes to the other daughter cell as one parent cell divides into two. Right? So when you see chromosomes in this kind of X shape here, this means it has been captured. The image has been captured at the time when DNA replication has taken place, and the two sister chromatids are still attached together as part of how they stay organized so that mitosis can properly happen. Right. I point this out because surely the general public, if you were to ask them, what does a chromosome look like? You know, they might be like, oh, I don't know, I guess it's those X's, right? We see those X's, right? And I think many life science students tend to think, yeah, the chromosomes in our cells look like these X's, right? So that can be kind of misleading because the reality is it's only for a brief time in the life of your cell that your chromosomes are actually arranged as these X's, right? Most of the time, they are not condensed like that. They are a mix of sort of loose strands of heterochromatin mixed with strands of just more relaxed loops. Okay. So we will talk about this further as we discuss the importance of chromatin organization in gene transcription or gene expression. Okay. So this then is a nice kind of summary cartoon that helps, I think, helps you appreciate these different levels of organization, right? We start with the DNA double helix, the naked DNA, right, that contains all of our genetic information. It wraps around histone proteins, the histone proteins that come together to form an octamer that we call a nucleosome, right? So about 146 base pairs of DNA are associated with the nucleosome. And even though the cartoon doesn't show it here, these histone proteins have their N-terminal parts of the protein kind of sticking out from inside the core, and those can be modified, right, to control how tightly or how loosely the DNA is wrapped around this. Right? So then this beads on a string begins to wind upon itself to form these coils. Right? And another way of thinking about it is you have these coils that form that can then also bunch upon itself as supercoils. So again, just think about the big rubber band that gets increasingly kind of jumbled up on itself, right? If you do that and you get this mass of a rubber band, the point is, right, the rubber band is still, one continuous rubber band, but it's now packed up in a very tight space. And that supercoiling then is what you see in a highly condensed chromosome that you would encounter when you're looking at a cell that's undergoing mitosis. So most of the time, that means in our living cells of our body that are not undergoing mitosis, that are just doing their jobs, the organization of the DNA inside the cells is somewhere between here and like here, right? And that means you would not see the chromosome as a condensed rod or an X. If anything, it would be a very long chromatin fiber mixed in with all the other long chromatin fibers that represent all the other chromosomes that you have in kind of this big mass inside of the cell's nucleus. So the last concept that I want to talk about today is going back to why the double helix structure of DNA is so important for providing mechanisms by which life can use DNA as an information storage molecule. Because throughout today's conversation, when I've been talking about how the organization of DNA is so important to get a huge 6-foot long, human genome DNA molecule down to a teeny tiny space of maybe just a couple micrometers in size, right? Very obviously, organizationally, that's part of why chromatin is important. But I also said, right, that chromatin is important because the cell can then selectively choose what parts of which chromosomes are unraveled. When the DNA is unraveled from there, you would have maybe little stretches of DNA that are naked, and then the base sequence of those stretches can now be read. The information is accessible. Right? So in order for the DNA genetic code to be read, right, the DNA strands actually have to be temporarily separated so that things like particular proteins can come in and actually read it. So this is one of the fundamental properties of why the structure of DNA is so important. Remember, the two sides of the ladder that make up the DNA, right, have their complementary bases pointing towards the middle, and the A's, the adenines, are across from the thymines, right? The guanines are across from the cytosines, and the bases pair through hydrogen bonding, the little dotted lines that represent a pretty weak interaction holding the ladder together. Okay? And I said before that relatively weak interaction is important, okay, because you need to be able to separate the two strands and have them come back together very dynamically. One way of separating these strands is just by putting energy into it, enough energy to cause the atoms that are involved in, for example, the base pairing, right, to vibrate around enough that they kind of lose the hydrogen bonding from each other. Right? That weak hydrogen bonding network can be broken if you get these atoms vibrating too much. So if you put heat, that's what heat is. Heat is you're putting energy into things to get molecules, to get atoms moving around. If you start off with a native strand of DNA in its normal double helical form, it'll stay double helical up until around 80 degrees Celsius. But if you heat it above that to like 90 degrees or so, right, that's too much vibration. The heat energy is vibrating all the nitrogenous bases too much, right? And then the two strands come apart. Importantly, though, if you then cool the DNA down and allow those atoms to kind of mellow out and slow down in their vibration, right, the two sides of the DNA ladder will come back together again because of that complementary base pairing, the A's across from the T's, the C's across from the G's. Right? And so you can then renature the DNA. Okay? So this is a very important discovery in the lab that's been known for many, many decades. You know, in your cells, in your body, of course, your cells do not separate the strands by heating them up to 90 degrees. We will learn that in our cells, we have particular proteins that act as little machines that carefully unravel the DNA where the genetic information needs to be read. But the fact that you can denature the DNA with high temperatures, and then you can rely upon the DNA strands coming back together after you cool it, forms the basis of some very important and widely used techniques in the lab. Those of you who are taking 275L this semester with Dr. Yoshizawa, you'll be using these concepts in the lab. And this is a technique that we will explore in Wednesday's lecture. Okay, so that's where I'm going to stop today.